Hello, my dear students. How are you doing? So, finally, your biology paper is done. And I hope all of you have rocked this particular examination. What you say? Yes. So, your examination was very, very easy. I have seen the paper, and literally, students, all the questions were discussed in your lectures, right? So let's see about the analysis, how your paper was, and we will also discuss the whole biology paper. So I and Rajni ma'am, your zoology and botany teacher, will be giving you the whole paper analysis with the proper solutions of the questions. So let us start with your biology paper discussion. So are you guys ready? I'm very excited. What about you guys? How many of you want to know or want to calculate at the end what marks you are getting in your biology paper? All of you will be very, very interested in knowing the answers so that you can recheck your answers and calculate your marks, right? Okay, so let us discuss about the pattern. All of you are already aware about the pattern because before going to the examination, you might have gone through the pattern of your biology paper. So over here, you can see section A included your 16 questions and each question was of 1 mark. So total 16 marks was for your section A, right? And section A was very, very easy. It was totally MCQ based, right? Then your section B included 5 questions and that too of 10 marks, right? That means 5 questions of 2 marks gives you 10 marks in your section B. Then section C includes 7 questions that is of 3 marks. So in total 21 marks. Similarly, section D and section E included 2 and 3 questions respectively, that too of 4 and 5 marks. So your overall paper was of 70 marks, right? So what I have analyzed, what I think your paper was, is written in this particular slide, right? I have written about each and every sections of your biology paper. So let us see what I have analyzed and have you also seen this particular thing in your biology paper? Okay, let us check that. So you can see over here, as I told you, direct questions were there from NCRT. All the questions were from your NCRT book. So we have already told you in all the lectures, all the faculties of your biology have already told you to go through your NCRT in detail. Nothing was out of NCRT, right? So that means paper was from your NCRT and direct questions were asked. MCQs that were your part A was highly scoring. It was highly scoring, right? Because all the questions were very, very easy. It covered both your theoretical as well as application based questions. Now, section B included all direct questions and was relatively very easier. So that means section A and section B was very, very easy. And all the students might have done all the questions correctly, right? We will see later on in the upcoming slides. In section C, the blend of theoretical, application-based and few analytical questions were asked of moderate complexity. So I can't say the paper was totally easy because in section A and section B, the questions were relatively easy as compared to section C. So we can say the paper was easy, moderate or tough. You tell me guys, what do you think? The paper was easy, moderate or tough? So I will say it was moderate. Not that tough and not that easy. Because the paper is comprising of all these sections? No. So we can't say it is an easy paper. It was a moderate paper according to me. Then in section C, case-based questions were asked that were again not that difficult to solve. Section E, that was the last section of your paper, included competency-based questions focusing on concept analysis. So from all these analysis points, I can say that paper included the questions from your NCERT and the level was moderate. We can say that yes, for average students, the paper might be challenging, but not that tough, right? So let us now check what were the questions in your botany sections and how you have done them or written them in your examination. So we will start with section A. As I told you, section A included your 16 questions, that too of 1 marks, right? So first question on your board is from botany itself. I will be dealing with botany questions and Rajni ma'am will take your zoology questions, right? Okay, so let us see what the question is asking you. Question says, in a fertilized ovule of an angiosperm, the cells in which N, 2N and 3N conditions respectively occur are. So this question is from your very own first chapter of class 12, that is sexual reproduction in flowering plants, right? So it is one of the easiest question. You have already done this type of question in your class, okay? So N, you have to tell about N, then you have to tell about 2N and 3N ploidy condition. Okay, so you know that all the cells that are present in an embryo sac are of haploid nature, whether it is antipodal cells, synergid cells, egg cells or polar nuclei, right? So here for N it is given antipodal, zygote, endosperms and antipodal. So zygote is 2N and endosperm is 3N. So definitely you can omit option B and option C. Coming to option A and D, antipodals is true for your first option, that is N ploidy. Okay. Now coming to 2N. 2N is your zygote. Absolutely correct. 2N is your synergids. No. Synergids are haploid in nature. So your option D is also false. Option D is also false, right? So what should be the correct answer for this question? Option A. N is for antipodals. 2N is for your zygote. And 3N for endosperm. Right? So answer for this question is option 1. Coming to the second question. Second question is from your chapter Principles of Inheritance and Variation. It is based on pedigree analysis. So you have to tell which pedigree it is from the given figure. So what should be the answer for this question? See, you might have learnt about various tricks to remember whether a pedigree is dominant or recessive in nature. From the figure, we can clearly tell that it is skipping the generation. So whenever generation is being skipped, it is considered to be a recessive type of pedigree. Okay, so it is not a dominant pedigree. So we can omit easily option B and option D. Right? So what is left with us? Option A and option C. So over here, if no generation is being skipped, then only we can say that it is a dominant type of pedigree. But generation is being skipped. How? Because parents are unaffected. Parents are unaffected and your children are affected. Some of the children are affected. So definitely it is a recessive type of disorder. Okay. Now in the option we have X-linked and autosomal. So how you are going to check? You know that in X-linked inheritance pattern or in pedigree we have criss-cross method, or it is also called as criss-cross inheritance. That means when a father is diseased, the daughters will be diseased. or when the mother is diseased or affected, all the sons will be affected. So over here you can see that son are also affected and daughter is also affected. So definitely it is not a crisscross inheritance. So option A is also wrong. So remaining option will be the right answer for this question, that is autosomal recessive. The answer for this particular question is option C, that is autosomal recessive. Okay, coming to the next question. I hope the two questions that we have discussed till now were very easy and direct from NCERT, and all of you have marked it right. Okay, let us see the next question. All right. The question is again very easy. Which was the last of the 24 human chromosome to be completely sequenced? This is the question from your chapter Molecular Basis of Inheritance. So it is the direct question. You have to tell out of 24 chromosomes, okay, 24 human chromosomes, which was the last chromosome that was sequenced. So it was chromosome 1 that was sequenced in May 2026, or you can say 2006, under Human Genome Project. Right? So answer for this question is option A, chromosome 1. It is the largest chromosome and contains largest number of genes. All right? Fine. So this is your next question from your section A. Very easy question. Directly the diagram of the NCERT has been taken and put over here, right? So this is the question again from your sexual reproduction of flowering plants chapter. So very easy. I don't think any one of you will face any difficulty in answering this particular question. See, over here A, B and C label have to be discussed. You have to tell what is A, B, C in this particular diagram. So what is this diagram? It is a diagram of young anther of an angiosperm. Same figure is given in your NCERT. Alright. So A is what? You can see A is present over here. It is a transverse section. That means the two lobes of anthers are connected by some tissues that are sterile in nature. So what those tissues are called? They are called as connective tissues. So A option is your connective. Means not A option, A labeling is your connective tissue. It is given only in one option, that is option A. So directly you can mark option A to save your time in your examination, right? Is that so? No, you have to just recheck your answers with the rest of the labeling too. Okay. So over here you can see option B. Option B over here is your endothecium, and option C, A is given as pollen grain, and in D option A labeling is given as endothecium, so all are false. B is endothecium, obviously B is endothecium, and you also know that C are your pollen grains that are getting released by dehiscence of anther. So your answer is option A over here. Clear? Okay. Coming to next question. Again very easy question. From which chapter this question is? This is from your Principles of Inheritance and Variation chapter. It is asking you about the Turner syndrome. Turner syndrome, it is a kind of chromosomal disorder that is present in humans. So what is the basic phenomena behind this particular syndrome? What happens in the human chromosome due to which this disorder or the symptoms of this disorder is observed or are visible in humans? So you all know Turner syndrome is represented as XO. 44 plus XO, this is 0, or you can write it as 44 plus X. That means one chromosome over here is missing. Chromosome is missing. Or you can say it is lost. So when one chromosome is lost, it is called as aneuploidy. Polyploidy always increases the number of chromosome. Euploidy also increases the number of chromosome, and it is not at all an autosomal abnormality. Why? Because it is asking you about the Turner syndrome, and Turner syndrome is seen due to the changes in the sex chromosome, that is your allosome, right? So correct answer for this seventh question of section A will be option A, aneuploidy. Okay, fine. Coming to next question. Again, this question is from molecular basis of inheritance. So far, whatever questions we have discussed, you can clearly see that most of the questions are from your principle of inheritance and variation and molecular basis of inheritance. So this has been already told you before your examination that these two chapters have very high weightage in your board examination. And that is what you can see in your paper that you did today, right? Fine. Coming to this question, very easy question, very easy question. What question is asking you? Which of the options has correct identification of P, Q and R in the illustration of central dogma given below? You know what is central dogma? DNA will convert into DNA by replication, DNA will convert into mRNA by transcription, and mRNA will convert into protein by translation. This is your central dogma theory. So over here you can say that DNA is converted into DNA by replication, then DNA is converted into mRNA with the help of process called as transcription. Very easy. Then what is this R? If protein is formed, this R will be your translation. So you get the answer over here. P is your what? It is not replication, not translation. It is transcription. Q is mRNA, R is translation. So answer is option D. Very easy, no? There should be no confusion in this particular question. All right? Very good. Coming to next question. Okay. So now we have completed the MCQs, means the option-based questions are completed now from section A. And you know that section A also included your assertion and reason questions too. So we have two questions from botany in your assertion reason part. Let us see what were those questions. Again, they were the direct lines from your NCERT. Okay. So you can see question number 13 over here. 13 to 16 questions were ANR, and you know these are the four options that you have to choose from to get the answer of these questions. So let us see what question 13 says. Question 13 is asking you about assertion, that is, a given fig species can be pollinated only and only by its partner wasp. This question is from sexual reproduction of flowering plants, and this is absolutely true. This is a true statement. Now let us check what reason is. Reason is given over here. The wasp pollinates the fig inflorescence while searching for suitable egg-laying sites. That is also absolutely true. That means both assertion and reason are true over here. So what should be the answer? Is it A or is it B? Because if both the statements are true, we can omit option C and option D. So obviously, this is the correct explanation. Reason is the correct explanation for your assertion. So, your answer would be option A. Assertion and reason are true, and reason is the correct explanation for your assertion. Fine. Very easy question. Direct lines from your NCERT. Next question. Assertion reason. From which chapter? Ecology, organism and population, and ecosystem. This question is asking you about some aquatic ecosystem have inverted biomass pyramids. Reason is more energy is required by the organism occupying the higher trophic levels. You know that in ecosystems you have already discussed and you already know that there are two types of pyramids or even three types of pyramids. You can have spindle shape as well. So there are different conditions in which we can have these three types of pyramids, right? Now, some aquatic ecosystems have inverted biomass pyramid. Here it is written some. Do you think it is correct? It is false. Why? Because it is not some, it is not mostly, it is all. So over here in place of some, all should be there, then only this statement should have been correct. But over here it is written some, that means there are some exceptions that do not have inverted biomass pyramid, and it is not true. In all the aquatic ecosystems, everywhere we have only and only inverted biomass pyramid. So this statement is false. What about reason? More energy is required by the organism occupying higher trophic level. More energy is required. Definitely the organism that is present at the upper level of this particular pyramid will have lesser energy. It will have lesser energy, but it will require more amount of energy to perform its function. So this statement is true. This statement is true, and above assertion statement is false. So if assertion is false and reason is true, you will have what? Option D. So for this question, your correct option is option D. I hope this question is clear to you all. Assertion says some should not be there. All aquatic ecosystem have inverted biomass. So this statement is false. Reason, more energy is required by the organism occupying higher trophic level. More energy is required, but they have lesser energy. Okay, so this is true statement. So this was all about your section A. Let us now start with our section B. Okay, so you have certain options in this particular section as well as in section C, D and E. Options were given in your certain questions. Okay, so first question of section B is, this is your section B. First question is, differentiate between grazing food chain and your detritus food chain. You have to tell the difference between grazing food chain and detritus food chain. Very easy. It is your 2-mark question. 2-mark question means how many points you have to write over here? 2 or 4? 2 points are enough. So you should write 2 pointers related to grazing food chain and detritus food chain. You know that grazing food chain starts from your grazers, okay, and it is mostly occurring in your terrestrial region, whereas detritus food chain starts from your dead decaying material or dead decaying the organisms who feed on dead decaying matter. So they are called as detritus. So DFC, detritus food chain, begins from your detritus, right? So this is the first difference. Second difference, they are the main energy conduits on terrestrial ecosystem and they are the main energy conduits in your aquatic ecosystem. So these are the two differences that are given clearly in your NCERT. Fine. So some might have done this particular question, some might have done this particular question. The second question is also very easy. It is asking you about brood parasitism. What is brood parasitism? And tell one example. So here you can just give definition of parasitism. You have to tell about parasitism. What is parasitism? You know, one is parasite over here, another one is your host. So parasite always gets benefit and host always gets harmed by the parasite. So this is your parasitism. So brood parasitism is what? Then you should write about brood parasitism. So brood parasitism is a special type of parasitism interaction where the parasite insect, or you can say not the insect, parasite bird lays its egg in the host bird nest. This is called as a brood parasitism, and it is a special type of parasitism. So first of all, you should write about parasitism, then brood parasitism, and then you have to give an example. So what is an example? You know that cuckoo bird lays its egg in in whose nest? Crow's nest, right? So this is an example of your brood parasitism, and it also shows a behavioral uh, pattern of certain birds. So cuckoo bird lays their eggs in the nest of crow, where crow doesn't give any parental care to the eggs of cuckoo. Okay, so this you should know. Okay, coming to next question, 21st question. So you have two options or two sub questions in question 21. A is asking you about biodiversity hotspots, covers less than 2% of the Earth's land area. Strict protection of these areas can reduce the rate of ongoing extinctions. Explain. You have to explain this. This is the line of NCERT. So you can clearly explain the causes of biodiversity loss. So in your NCERT, the topic is given loss of, topic is given in your NCERT chapter of ecology, loss of biodiversity. This is the question from your biodiversity and conservation chapter. So this is the topic of the NCERT. You have 4-5 points written over there. You can choose any 2 points and write in this particular answer. Very easy. It is asking you how this biodiversity loss is occurring. So it is occurring due to certain reasons. So 4 pointers are given in your NCERT. You can write over here. Alien invention, and you can say the habitat loss, right? So 4 pointers are given in NCERT. You can mention any two. Next part of this question is asking you about, name any two hotspots in India. You have to write the hotspots in India. You know that. You can write the Himalayas, and what you can write? What you can write? Western Ghats. So many examples are given in your NCERT. You can take any two and you can mention over here. I hope this question is very easy and you guys faced no issue in answering this question. It was a direct question. You have to just mention the pointers of loss of biodiversity and two examples in hotspots of India. So that was all for your section B. Coming to section C. So quickly we have to see what was your question number 22. Okay. So question is asking you about, draw a labeled diagram of sectional view of male gametophyte or microspore of an angiosperm and write the functions of any two parts labeled. Means whatever diagram you are making, diagram of male gametophyte or microspore, you have to label 4 parts from that and you have to mention the role of only 2. Don't write the role of each and every labeled section. Okay, only 2 roles. Sorry, 2 labeled part roles have been asked in this question. So kindly read the question very carefully. Alright, so let us draw. So you have to draw the male gametophyte. Male gametophyte is what? It is a pollen grain. And you know, pollen grain contains 2 layers. So you should draw an intine and you should draw exine. Okay. So this is your intine. This is your exine. So these are the 2 labeling. Then what you can draw? You can make two cells over here like this. You can make two cells over here like this. So let me make one larger. One large cell and one small cell. One large cell, one is small cell. Why I am making one large and one small? Because this one will be your vegetative cell. This is your vegetative cell. And this one is your generative cell. Right? So these are the four labelings. 1, 2, 3 and 4. That's it. No need to mention any other labeling of this particular part. It is asking you about the male gametophyte. Male gametophyte is none other than your pollen grain in angiosperm. So you know that it have inner layer intine, external layer exine. Then you have two cells. It is a two-celled stage condition. Two-cell stage. Right? So one is bigger cell called as vegetative cell. Another one is a small cell called as generative cell. You can write functions of any of these two. Okay. Exine gives protection. It is made up of sporopollen that is resistant to your alkali, your pH, enzyme. Everything is written in your NCRT. Vegetative cell is a larger cell that provides nourishment to gametophyte, to the generative cell and the gametes, right? Then generative cell leads to formation of two male gametes. Okay, and it is smaller in size as compared to vegetative cell. So these are the various functions of the labelings that you have done over here. I hope all of you have drawn this thing only in your examination. Okay, this is your pollen grain, this is your male gametophyte. All right, coming to next question, 23rd question. This question is again from your principles of inheritance and variation. Very easy question. It is an analytical question. You have to read the question carefully, then analyze what it is saying and then write it down and make a Punnett square. So what the question is asking you? Question is asking you, a man with the blood group A marries a woman with the blood group AB. So man is having a blood group A that marries a woman with blood group AB. So these are the genotypes. Then the first child born to them has a blood group B. So first child has blood group B. So this B would be coming from the mother. This is your mother. This is your father. So this IB is coming from here. So obviously this I might be coming from father. So this is the genotype of your firstborn child. Now the question is, work out a cross to find the genotype of the father. Give the possible blood groups and their genotype of the children that could be born to this couple and use Punnett square. So the answer for the first question I have given you. Father genotype would be I, A and I because the blood group is A. So this is the answer for the first half of your question. Second half is asking you, work out the cross to find the genotype of the children that could be born to this couple. This couple, only one child is given over here, one child genotype is given over here. You need to calculate the genotypes for rest of the three child. So what you have to do? You have to make a Punnett square. It is very easy. Just make a Punnett square. So in Punnett square, what you need to do is first mark the male and the female gametes. Okay. So suppose this is male gamete, this is female gamete. So in male gamete, you have I, A and I. Here in female, you have I, A and I, B. Now you should make the combinations. So combinations will be I, A, I, A, I, A, I, I, A, I, B, I, B and I. So these are the four genotypes of the children that are supposed to be born from these two parents. Okay. So first child is this that were given in the question. rest of the 3 childs will have this particular genotypes. So like this, you have to make a Punnett square in your paper and answer this question like this only. Okay. Coming to the second part of 23rd question. State the basis of ABO blood grouping in humans. What is the basis of ABO blood grouping? You know that it is multiple allelism. You have to just write this word. Multiple allelism or multiple alleles. You know that only one I gene is there that have 3 alleles. IA, IB and IO. You have to mention only this much. So this was the answer for this particular question. Okay. Coming to next question, that is 26th question. It is asking you, explain the role of the following during the sewage treatment. Very easy question. Very, very easy question because you were already told that this particular sewage treatment topic is very, very important for your board examination. And see, the direct question has been asked. And that too of your 3 marks. First is you have to write the role of flocks and you have to write the role of anaerobic sludge digester. So 1.5 marks for this and 1.5 marks for this. So you have to write 3 points. Only 3 points you have to write. So what is the role of flocks? You know what are flocks? So you should write that they are the mess that are formed of fungi and certain bacteria. That is the mass of bacteria. So the mass of bacteria looks like a mesh of fungi. That is a flock. And where they are going to play an important role. They play an important role in your primary treatment or secondary treatment. Secondary treatment, that is biological treatment. They help to reduce the organic matter content present in the water. है ना? That means they are helping to reduce BOD, reducing organic matter content. Okay, so you have to write this role of flocks. Then anaerobic sludge digester. So anaerobic sludge digester, what is the role of this? It helps to digest the activated sludge. Activated sludge. It includes anaerobic conditions that helps to digest all the bacteria and all the fungi present in the digester. So these are the roles given in your NCERT directly. So there should be no issue, no doubt in previous question. Okay. So coming to section D, this is the second last section of your biology paper. So let us see what type of questions were asked in section D. Okay. So it is very easy question. It is based on figure. This figure is from your ecology unit and the same figure is given in your NCERT. It is telling you about the population curve, right? Or you can say the growth curves, not the population curve, growth curves. Okay. Now the question is asking you about A and B. What is A? What is B? You have to label these two curves. So let us read the question. Populations evolved to maximize their reproductive fitness in the habitat in which they live. Ecologists suggest the life history of organisms have evolved in relations to constraint imposed by biotic and abiotic components of the habitat in which they live. This gets reflected in the population growth pattern of all the organisms, including humans. So study the population curve in the given graph and answer the following questions. First, identify the growth curve A and B. So can you tell me what is A and what is B? A is your exponential growth. Exponential growth. And B is your logistic growth. The same graph is given in your NCERT. There should be no dikkat in solving this particular question, right? So why it is called as exponential growth? Because you can see the graph is going up and up. That means the resources are unlimited in this particular population. So as much as you are giving the nutrients, you are giving the resources, the population size is increasing. Whereas in logistic growth, the nutrition or the resources are limited. So population size is controlled in logistic growth. So answer for first question is exponential and logistic growth curve. Okay. Next is mention what does the dotted line in the graph indicate and state its importance also. So two questions have been asked in your option B. What is this dotted line and what is its role or why it is very, very important. So what is K? You know K is your carrying capacity. K is your carrying capacity. Okay. K is your carrying capacity. Carrying capacity. So the first half answer will be carrying capacity. And why it is important? You all know that if the resources are limited, they are not unlimited for a population. So definitely one population can have only a specific number of individuals so that it can provide its proper nutrition and proper growth can occur, right? So that's why the number of individual that a population can sustain is called as a carrying capacity. Okay, so section B, sorry, the option B answer would be carrying capacity, and I have told you why it is important. Okay, so you can see in this particular question or is given. That means you can answer this question or you can answer this question. And both the question are from graph only. So let us see what the next question, optional question is asking you. Again, it has two parts. And it is asking you, growth curve B shows a different pattern from that of growth A. Justify giving one reason. So I have already told you, exponential growth have unlimited resources, B have limited resources. That's why it can sustain a specific number of individuals in them. So the answer for first question is clear. Now, second question have two parts. First is, which one is more realistic? Obviously B1. Why? Again the same thing. They are asking the same things again and again just by modifying the words. The answer remains the same. Why? Why B is more realistic? Because in nature, the resources are not unlimited. They are limited, right? That's why a lot of biological interaction occurs like competition, parasitism. So due to the limit of the resources that is provided by the nature, the logistic growth seems to be more realistic. Next part, which one of the two curve is relevant in present days with respect to human population in our country and why? Again, the B. The reason remains the same. So B has stole all the light of the show, you can say. The answer for all the options, all the questions will be B, logistic growth over here. Okay, very easy question. Fine, coming to next question. Read the following passage. A passage is given to you and certain questions have been asked from you. What is the passage saying? Generally, in eukaryotic cells, the average length of a transcription unit along a DNA molecule is about 8,000 nucleotides. So, the RNA produced by transcription is also that long, 8,000 nucleotides. This is what? This is your DNA sequence. Okay. Then it is getting converted into mRNA and it is also having the same length, 8,000 nucleotides, right? But it only takes about 1200 nucleotides from the above RNA product to translate average size of polypeptide. Okay. Only few part undergoes to form protein. And the number given over here is 400 amino acids. Okay, fine. So the question is, name this RNA product transcribed from the DNA that subsequently translate into polypeptide of this. So obviously you know that initially this mRNA is not formed. You know that what is formed is your hnRNA. hnRNA. This hnRNA undergoes certain processing and then it leads to formation of this mRNA. mRNA is your messenger RNA, right? So directly your this 8000 nucleotide will not undergo the formation of mRNA. After certain processing like splicing, your adenylation and your tailing and your capping. These are the three processes through which hnRNA is being processed and it leads to formation of mRNA. So that means this mRNA is the thing that is being asked over here. So answer for first part will be mRNA, because whole 8000 nucleotide are not undergoing the formation of protein. So some part will be lost during the processing process and mRNA will be formed that will lead to formation of protein. So answer for the first part is your mRNA. Mention the enzyme responsible for transcribing this type of RNA from the DNA. From DNA, this RNA is produced. You know, during transcription process, RNA polymerase is required, right? So answer will be RNA polymerase. Okay, RNA polymerase 3 you can write over here. Okay. Next is name and explain the process the RNA molecule transcribed from 8000 nucleotide long DNA undergoes to translate polypeptide of this. Okay, you have to name and explain the process the RNA molecule transcribed from this. So you can write the three methods of processing that are called as post-transcriptional modifications: splicing and your tailing and your capping. Okay, so you have to write about these 3 pointers over here. Third is write the number of RNA polymerases involved in transcription of DNA in pro and eukaryotes. You know that in prokaryotes we have only 1 and in eukaryotes we have 3. Okay, 1, 2 and 3. So this is the difference. You have to write only the number. No need to write the names. Okay. Or again you can see this question has two options. You can write answer for this question or you can write the answer for this question. So option A and B are mandatory of this question, and in option C you have the optional questions. You can write either this or this. So second question is mention the difference in the site of transcription in a prokaryote and eukaryotic cell. What is the difference? You know that in prokaryotes, transcription and translation both occurs inside cytoplasm only, right? So you can write in prokaryotes, it is cytoplasm. Okay? Transcription it is asking. And in eukaryote, it occurs in nucleoplasm. You know that in eukaryotes, we have two different nucleoplasm. You know that in eukaryotes, we have a specialized mechanism where transcription and translation occurs in two different locations. But in prokaryotes, they are very simple cells. They do not have complexity as that of eukaryotic cells. So transcription, translation both occurs in cytoplasm. So you have to mention these two. So A, B are common questions for question 30, and in C you have an option. So previous question was very easy question and it was the direct question asked from your molecular basis of inheritance. Okay, coming to next question. This question is from your section E. Okay, and over here you can see one figure is given to you and certain questions have been asked. So these diagram based questions are very easy because the diagram are from your NCERT only. Direct diagrams from NCERT has been taken. This question is from sexual reproduction of flowering plants. So let us see what the question is asking. Observe the picture of Commelina plant bearing two type of flowers given above. What are the two type of flowers? Option A, option B. Two labeling has been done in this particular diagram. You have to tell what is option A, what is option B. You know two type of flowers are there. Chasmogamous and clistogamous. Chasmogamous and you have clistogamous. So what is the difference? Chasmo means open flower, you know that, and clisto means closed flower. So these are the two type of flower that are shown over here. So obviously the flowers that are present inside the soil or underneath the soil will be your clisto ones. Clisto means close. Okay. And the ones that are present above, they are called as chasmo. Chasmo means open. So A and B are chasmo and clisto. Second part is compare the two type of flower with reference to characteristic feature and mode of pollination. Very easy. I have told you about the characteristic feature. One is open flower, one is closed flower. And what about mode of pollination? You know that mode of pollination for chasmogamous will be, as it is an open flower, so it will be cross pollination. Okay. Whereas clistogamous is your closed flower, so it will undergo only and only self-pollination. So you have to write only about this much. And if you want to write in detail, you can write about autogamy, geitonogamy and xenogamy, but it is not required over here. Okay. Third option is list any two outbreeding devices. In flowering plants, explain why do plants develop such devices. So you know that self-pollination is not favorable for the plant, so they undergo or they want to have cross-pollination. For that, outbreeding devices are needed. So I think three or four types are given in your NCERT, that yes, plants can have this type of method as an outbreeding devices to have cross-pollination. So it is written in your NCERT, in your sexual reproduction in flowering plants, under the heading outbreeding devices. So you can take any of the two devices and mention over here. Direct question from NCERT, no issue in this question, right? Very good. Okay. Very interesting question again from molecular basis of inheritance. You have one diagram that is showing you the step of translation. You can clearly see over here translation is being shown, right? Okay. So what is happening over here? You have one growing polypeptide chain. This is an exit site. You have an P site, you have an A site, you have mRNA, you have ribosomes. Okay, let us see what the question is asking. Question is asking, from the figure you can answer these part or you can answer these part. So let us see from the top what is option 1. One part is asking you, identify the polarity from X to X dash in the mRNA segment shown in the figure. Okay. X and X dash, this and this. So you know it is 5 prime and it is 3 prime. Very easy. Next, mention how many more amino acids can be added to the polypeptide chain that is being translated and why. So let us see what is present over here. So you can see at the end it is written as UAA. So what do you mean by UAA? UAA means stop codon, right? So when at the end of your mRNA sequence stop codon is present, that means at this particular point your polypeptide chain synthesis will stop. Synthesis will stop. So you can't add any of amino acid at this particular point as it is a stop codon. So answer will be, how many can be added? 0. Why? Because it is a stop codon. So the protein synthesis will not occur. You know that stop codons UAA, UAG, UGA, UAA, UAG, UGA stops your what? Polypeptide synthesis, right? So it will not add any of the amino acids. Okay, coming to the next part. Write the initiating codon for translation and its anticodon and the amino acid it codes for. Initiating codon, you know it is AUG, that is 5 prime to 3 prime. So its anticodon will be A will change to T. No, it will change to what? U. U will change to A and G will change to A. Okay. And it is written the amino acid it codes for. This codes for your methionine. Methionine. Okay. This is your mRNA, right? Okay. So A will code for U will code for A, and G will code for C, UAC. It will be C. UAC. Okay. Yes. So this will be your initiator codon, that is coding for methionine. Explain the charging of adapter molecule, why this molecule needs to be charged. You know that it is very important to charge an adapter molecule, that is your tRNA, because it brings a specific amino acid to the mRNA codon. And each and every codon present on mRNA have a specific tRNA except your stop codons. And you know the process of charging of an adapter molecule. You know, amino acid first gets attached to ATP, it gets activated, then it's get attached to tRNA, right? So amino acylation of tRNA is the same process of charging of an adapter molecule. So you can write the steps that are given in your NCRT clearly. Okay. Next or optional question was, why is sickle cell anemia a human blood disorder so named? It is a blood disorder where the RBC changes its shape. You know that RBC has biconcave shape, right? Biconcave shape. But due to sickle cell anemia, it changes to sickle shape. Now the question is asking, why is sickle cell anemia is named so? Due to the change in the shape of RBC, it is named so. Next question is, explain the genetic basis that results in the expression of this disorder. What is the genetic basis? So you have to write the codon thing. You know that at the sixth position in your polypeptide chain, glutamic acid get converts into valine. So certain figure has been given in your NCRT. You have to mention the same thing. How DNA is converted into mRNA and how in that mRNA the single nucleotide base changes and lead to change in the shape of RBC. It is given in your NCRT. You can take reference from there and you have to write the same genetic analysis of sickle cell anemia from there. Right? Okay. Next is, work out a cross to explain how normal parents may have a sickle cell anemic child. So what you need to do is, over here you have to write normal parents. So you know normal parents will have this type of condition, right? This is a normal condition and this is also your normal condition. This is the normal condition of an individual. But HB, but HBA, sorry, HBS will be your affected individual. So what question is asking you? Work out a cross to explain how normal parents can have a sickle cell anemic child. So what you can do is you can make a cross between HBA, HBA, HBA and HBS. Both are normal individuals, as I have told you. So what will happen when the gametes will be formed and when the children will be born from these two parents? One will have HBA and HBA. One will have HBA, HBS. One will have HBA, HBA. And another will have, see over here you have to write HBS. Okay. So don't write like this. You can make a Punnett square. Okay. Make a Punnett square like this. So what you will have? You will have HBA HBS. Okay. And over here, what you will have? You will have HBA and HBS. So both are your normal parents, right? So what happens? HBA, just give me 1 minute. You will have HBA, 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 HBS. You will have HBA, HBS, and you will have HBS and HBS. So what parents you should take? You should take heterozygous parent. These are heterozygous parent. That means they have some of the genes that can cause your sickle cell anemia in the coming generation, but they are not affected. They are normal individual. This is normal. This is normal. Okay. So when you will cross them, when you will cross the gametes from them, male and female, you will get one child as diseased. This child will be your sickle cell anemic child, right? So this is an affected child. This is your affected child. Okay, and these are your normal child. You can say these two are carrier. These two are carrier and this is normal. Okay, so this is how you have to draw a Punnett square across to explain when two normal individuals, normal parents have a sickle anemic child. So it is only possible when parents are heterozygous in nature. Why? Because sickle cell anemia is your what type of disorder? You know the pedigree, right? So the parents has to be recessive. Sorry, the parents have to be heterozygous in nature. They should not be homozygous. Okay, so if they are heterozygous, so one child definitely will be affected and will have sickle cell anemia.
So let's discuss about the zoology portion from the set 1 biology board exam. So this is the question. So the question is, select the option that gives the correct identification of ovum, morula and blastocyst. Direct NCERT diagram and question, right? So we know A is ovum, agree, A is ovum, and then when we talk about this is E, right? So they are asking us what is A, what is E. So E is the morula. This is morula, right? And they're asking us to label the blastocyst. So G is the blastocyst. So you have to choose the correct option. So this is the NCERT diagram. They have labeled all the things as an NCERT, right? So what will be the perfect answer? Obviously, A is ovum, E is morula, and G is blastocyst. So C is the perfect answer here because this is the diagram from NCERT and you have to give the correct answer. Super easy. Got it? Any doubt? You can just shoot it in the comment box and I will be super active today. I'll be, interacting with you guys in case you have any doubt. So let me take you ahead. So now the other question is the pills. You have contraceptive method here. What is their mode of action? So the pills will prevent ovulation because they have hormones, progesterone or combination of progesterone or estrogen that we have done in the class. And we know that progesterone is anti-ovulatory hormone. So that means it will prevent ovulation, right? So it will go, this goes with it. And then when we talk about this condom, it will prevent sperm reaching cervix. Vasectomy, in which semen means this vas difference is cut and tied so that sperm is not going out. So semen is coming out without any sperm. So correct answer will be this. Copper T prevent implantation. That's true. So A goes with 3, B goes with 1, C goes with 4, and this goes with second. So correct answer will be C. Isn't it easy, right? So this is directly from NCRT, and we already have done it very nicely. You might have studied your NCRT, the proposed mutation theory. Who proposed mutation theory? Super easy question, right? So Weismann actually disapproved this Lamarck theory, right? By just giving the germplasm theory. When we talk about Charles Darwin, he has given the natural selection. And Louis Pasteur has done this experiment to show that life comes from the pre-existing life, right? And Hugo de Vries has given the mutation theory. So correct answer will be D, absolutely D, right? Next question is the molecular scissors fall in the category of dash. So we know that molecular scissors are restriction enzymes. So out of all, this is making the correct sense, right? So that means this will come in the fall in the category of restriction enzyme. So why do we call it restriction enzyme? So we have done in the class because these molecular scissors, right? So they will be restricting, or they recognize a restriction site. They have the recognition site on DNA where they will cut it, right? And why do we call it restrict enzyme? Because we know that in bacteria, these molecular scissors, these restriction enzymes are already present, naturally present in their defense. So if anyway viral infection happening, or if virus trying to infect the bacteria, we know these restriction enzyme will restrict the growth of virus particle, viral DNA. It will not allow viral DNA to incorporate with the bacterial DNA. So they are restricting the growth of bacteria of the virus in the bacteria, right? So from there this name came, that is restriction enzyme, right? And obviously this molecular scissors is broadly comes in the category of restriction enzyme. Another question is, ELISA technique is based on the principle of antigen-antibody interaction. Easy. Antigen-antibody interaction. So correct answer will be the B. Antibody-antigen interaction means, suppose a patient having some disease, right? So that disease has certain type of antigens. So we will just isolate those, cells, or some kind of particles from their body, right? Or blood sample from their body, and we will just, suppose we are having the doubt that this person might be having this type of antigen. This person might be having this disease, so they must have this antigen. So we will use the preformed antibodies, or we will use the antibodies for that, corresponding antibodies for that antigen. So if that antigen interact with that antibody, if those antibody and antigen interact with each other, that means that person, it is confirmed that that person having this antigen which we were suspecting. So in that ways our doubt will be cleared. So generally ELISA means enzyme linked immunosorbent assay. So immunosorbent means we are just playing with the antigen antibody, right? So that means antigen antibody interactions is so crucial in the ELISA. So we just check for the antigen or a specific antibody in the patient body, right? Next is the assertion reason question. Plasmid are autonomously replicate circular extra chromosomal DNA. That's true. And plasmid are usually present in eukaryotic cells. So what do you say? So correct answer will be plasmids are usually present in not eukaryotic but in prokaryotic. So this is false, this is true. So assertion is true and reason is false. So you will choose the C option. So which says that assertion is correct but reason is false, right? So correct answer will be C in assertion reason, right? Next question. Patents are granted by the government to an inventor. That's true. So that other people cannot misuse that invention, right? So that because prevention, it prevents others from the commercial use of the invention. That's true. So correct answer should be A, that assertion and reason both are correct, and reason is perfect for the assertion, right? So if we use because patents prevent others from the commercial use of invention, so that is the purpose, right? Next is this graph. So you have to study the graph and you have to represent what does it mean, what is the X and what is Y. So this is the wall of thickness of uterine wall, right? So we know that uterine wall thickness keep on changing with the menstrual cycle. So it will grow in the proliferative phase, Yes, that's true. And in the luteal phase and when there is no pregnancy, it will go down. So this is the normal menstrual cycle graph. So it represents absence of pregnancy. That means there is no pregnancy. And absence of pregnancy means now progesterone level has gone down and wall will break down to form the menstrual bleeding, right? So it will cause the menstrual bleeding. So it represents menstrual bleeding. But when we talk about X, means this wall become more thicker after in 29, 20 days or 30 days. So means it's just getting so much thick. So it is showing that now female is pregnant, and that's why progesterone level is high. And when progesterone level is high, obviously thickness of the wall, uterine wall is maintained. So it shows pregnancy because wall is getting thicker, right? Pregnancy. It shows pregnancy in the female. So simple, you have to tell what is X and what Y indicates, right? So give suitable reason, because you know that absence of pregnancy means corpus luteum will degenerate. Hence, progesterone level will go down, and progesterone is the root cause. When the progesterone level fall down, that is the root cause of menstruation bleeding. So that means whole uterus wall is shedded off, right? And when the wall is getting thickened up, right? So it is getting thickened. So that means progesterone level is already high in the body, even after 28, 29 days. That means female is pregnant now, right? So that is the indication. Intense lactating mother generally don't conceive. Why? Because during intense lactation, which is after parturition, right? So during intense lactation, we already have done during intense lactation what happened. So during this time cause lactational amenorrhea. So this is amenorrhea. So what is amenorrhea? Lactational amenorrhea means absence of menstrual cycle. Why? Because and it is effective up to 6 months, right? Why menstrual cycle is absent? Because during this lactation time, prolactin hormone is high. So level of prolactin is so high. And when the prolactin level is so high, it will prevent the secretion of LH and FSH. So when there is no LH, FSH, no ovulation or no menstrual cycle. Because LH and FSH is responsible for the estrogen and progesterone secretion. You know how. So in the menstrual cycle, we already have done that LH, FSH act on the ovaries, and ovarian hormone, estrogen, progesterone act on uterus. Right? So ovarian event is basically controlled by this. LH is causing ovulation, FSH is maturing follicles, and if it's not happening, estrogen, progesterone will not be secreted, and hence menstrual cycle will be absent and no ovulation happening this time, right? So means at this time, no ovulation, and this is effective up to 6 months maximum, right? Or no menstrual cycle, hence no pregnancy, and that's why this is happening, right? That's why the female cannot conceive. Our government has intentionally imposed strict condition for MTP. Why? So you guys know very well that some people are so cruel. So they they go for this amniocentesis process. They get the sex of the child or a fetus, get the sex of the fetus checked, and then they are going for the female feticide, right? So these are the cases are reported high in the India, female feticide cases, right? So to prevent the misuse for the female feticide, or to prevent this discrimination, they have just intentionally imposed some strict action towards the MTP. So a person can go for MTP under certain condition only, right? So what is the reason why this is intentionally having the strict condition on MTP in India? So why? I have already told you, because people were misusing it, right? So to prevent misuse, to prevent the misuse of MTP for female feticide, agree? And the other reason can be like to prevent this, to check in this discrimination. So that is the reason it is having some strict action, right? So it is having some, imposed strict condition on MTPs. Next question. So I'm sure that everything is directly from NCRT. You might have found them super easy today, right? So write down in the comment box, are you finding them easy? Name the source from which insulin was extracted in earlier times. And why is this insulin no more is used by a diabetic patient? Because we are taking this protein, this is the proteinaceous hormone, insulin, right? And it was causing the allergies in some patient or some immunological actions in their body. So that's why we just stop using the insulin from the pancreas of the slaughtered animals, slaughtered cattle or pigs. So these were the sources of the insulin earlier. We were taking the insulin from the slaughtered, means those who are who are butchered, right, slaughtered. So from their pancreas we were extracting insulin and injecting it in the patient. But many patients have developed this allergy, right? So from source is from pancreas of slaughtered. So this is how you have to write down the answer, right? Slaughtered cattle and pigs. So that means what is the sources? Animal is the source, was the earlier source for this, what we called the insulin, right? And we have stopped it. Why? Because some patient was showing the allergy. Some patient develop allergies because they were showing allergic reaction, or they were showing some other type of reaction to the foreign protein, right? Or other reaction by their body, by their immunity, other reaction to the foreign protein. So you know that insulin is the proteinaceous hormone, right? So their body was showing this allergic reaction toward this foreign protein, and that's why we stopped them using. We are not using them for this, by the diabetic patient now. So why does the insulin synthesized in the human body undergo processing, whereas the insulin produced by Eli Lilly company doesn't need to undergo any processing? Explain. So how? I'm sure that you have explained it beautifully. Naturally in human body and artificially, which is formed by Eli Lilly company, right? So they have produced this genetically engineered insulin, also known as humulin. So how they have created it, and in human, what exactly happened? So we know that human has this, structure of this, what we called insulin, right? So this is the insulin structure. These are the sulfide, disulfide bond. And this is the proinsulin. This is the proinsulin. Proinsulin means inactive insulin. It has A chain, it has B chain and C chain. This is how it is synthesized in human in inactive form, right? And then, in our body, C chain is removed, and then later A and B chain are basically come together. They are forming the disulfide bond, and C chain is removed from the mature RNA, from the mature insulin, right? So this C chain is removed. So you can see this C chain is removed. So this is the mature insulin. So mature active insulin has only A and B chain, and this is how the processing happen in the human naturally, right? So humans secrete this insulin in inactive form, and it get activated only when this C peptide chain is removed, right? So this is what happening in our body. This is how the processing happening in our body. But when we talk about Eli Lilly, so they have written beautiful line in the NCRT that what exactly they have done. They have artificially, first of all, they have studied the sequence of the nucleotide in the gene of this A peptide or gene of the B peptide. So they have studied the gene which is responsible to form A peptide and gene in the human responsible to form the B peptide, because they never wanted to produce C peptide, right? So they artificially created, as per our NCRT, they artificially created the gene which is responsible for A peptide and gene which is responsible for B peptide, right? So these are the two genes. They have artificially produced them. Then they have introduced this gene in the host, and host was E. coli, right? So they have introduced this, what we called this A gene separately in one E. coli, and they have introduced this B gene separately in the B. E. coli, means in the other container containing the other E. coli set. So they have taken the two mediums, right, separately. They have taken the two mediums. In one medium, they are inserting the plasmid of the E. coli, which is the vector. We know that plasmid is the vector, which is carrying the gene for A peptide gene, right? Which is carrying the gene for A peptide. And it is carrying the gene for B peptide. Agree? And now they will synthesize the A peptide and B peptide chain. So these E. coli producing the product, which is A peptide, and they are producing the B chain. And then finally, when in two different conditions, in two different containers, they have produced A chain, B chain, they have separated, isolated, downstreaming it. And after the downstreaming, they have pasted A and B chain with the disulfide bond. So this is A chain, B chain. Later they have connected, linked them using disulfide bond, and they have produced directly this insulin, also known as humulin. So this is how they have produced the genetic engineering insulin, right? So that means this is not required. They have not produced, they have not taken the gene which is responsible to form the C peptide. They directly form the gene. They artificially created the gene which is responsible to form only A peptide and B peptide. So if you have written your answer this way, beautifully, comparatively, then obviously you can write down answer in the point form also, right? So this is how you must have presented it in the class, because I have given you it in the short notes in our YouTube channel. If you have seen, I have given you this comparison also, right? So now the other question is, whose skull ABC? But this is NCERT diagram. So A is the structure of adult human skull. This is the baby chimpanzee. This is the baby chimpanzee. And this is the adult chimpanzee. And they're asking you that which one looks similar. So you can see that baby chimpanzee skull looks similar to the adult human skull, right? So they are having similarity. But adult chimp, have different type of skull, right, than the human. So they are showing the similarity. So A and B are similar. They are showing similarity. And now they are asking you that name the dash ape-like and two man-like primates that existed 1.5. No. So they have written the wrong data here. So in our NCERT, it is 15 million. But here they have written 1.5. But that's fine. We neglect it. So this is directly NCERT line. So which one is ape-like? So when we talk about ape-like primate, it was Dryopithecus. So Dryopithecus were more ape-like, right? And then when we talk about the other primate, which were man-like, so it was Ramapithecus, directly NCERT line. So yeah, they were man-like, right? So this was your answer, but they have given this 1.5 million years ago they were existing. As per our NCERT, they were existing 15 million years ago, right? So there is some mistake, error here. But don't worry, right? So they have asked you ape-like or man-like. So it was direct question from NCERT line. So isn't it easy? What do you say? Yes, this is directly NCERT diagram. So this diagram is from drugs part, cannabinoids. So yes, this is the cannabinoids. How are such drugs consumed? They are either taken orally, either the people are ingesting them orally, or inhaled. They are inhaling them with the help of nose, right? And other one is name the human body organ affected by the consumption of these drugs. So it is affecting cardiovascular system. That means heart and blood vessel, right? It is fluctuating BP, heart rate, heartbeat. So it is fluctuating these organs, no? And the receptor of these cannabinoids present in your brain, in your central nervous, but they are asking you which organ is affected by consumption of this, right? So if we talk about the affecting the organ, it will be cardiovascular system, no? Another question they have asked, you have to draw the antibody molecules, which is having the 4 parts, two heavy chain, and 2 light chain. So the formula is H2L2. And we know that this is the diagram from NCERT. So this is the heavy chain, and this is the variable region of light chain. This is the constant region of light chain. This is the variable region of heavy chain, constant region of heavy chain. This orange color is the constant region, right? So why do we call them variable? Why do we call them constant? I already have, given you everything in our short notes in the YouTube, right? In Physics Wala Gulf channel. So I'm sure you have gone through those notes once, right? So yeah, so when we see the variable region of light chain and heavy chain, between these two structure, between these two variable region of heavy and light chain, we have antigen binding site, where antigen, it bind with the antigen here, right? It bind with the pathogen antigen over here with the help of this structure known as paratope, right? So these are the paratope where it is binding with the antigen. So this is the structure. You were supposed to draw this structure, and you were supposed to label it very, very nicely, right? So you will be writing that this H2L2 means they have 2 heavy, 2 light chain. They are bind with the, they are, stay together with the help of disulfide bond. So you might have written all the points in your exam, right? Name the cells which produce them. So we know B cell, B lymphocyte are responsible to produce antibodies. So we already have done that B cell are responsible for humoral mediated immunity, antibody mediated immunity. So B cell has this role, right? Or if you really want to explain it in more detail, you can write down all the things that I have told you in the class. I'm sure that you might have written the answer like how the B cells secrete the antibody. So in that ways also, but yeah, they are asking you only the name. So you can write down the diagram, some points of antibody that I have told you, and then you directly can write down the B cells secreting the antibody. Hence, it is providing antibody mediated immunity or humoral mediated immunity, right? So this is the diagram. Okay, so this question is from biotechnology PCR technique. So we know that it is having 3 steps. What are those 3 steps? One is denaturation at high temperature. Denaturation at high temperature, where these 2 DNA strands are getting separated. Second, after denaturation, we will be annealing. Annealing means pasting our primers. So you know what is primers? Single-stranded oligonucleotide, right? So that will be binding with the at the 3' end of both the template strand. And C is extension. So we know the third step will be extension, right? So B are nothing but the primer. And primer are the small segment of the small single-stranded oligonucleotide, which are getting pasted at one end, and they will initiate the replication, right? They will cause the extension with the help of DNA polymerase enzyme. So here, what is C? C is the Taq polymerase enzyme. So we know we use special type of DNA polymerase, which is the stable at high temperature and extracted from the Taq Thermus aquaticus bacteria. This enzyme is extracted from the Thermus aquaticus bacteria, which is the thermostable bacteria. So you have to identify A and D. So A is denaturation, D is extension. What is B? B is the primer, which are the single oligonucleotide, and they will initiate the replication. They will be annealed at the end, at the 3' end of the template DNA strands, right? What is C? C is the Taq polymerase, and name its source of organism, Thermus aquaticus bacteria. These are thermostable bacteria living at very high temperature, living in volcanoes, right? So their enzyme will be stable at very high temperature, because in PCR we are using high temperature, right? This PCR will be, these steps will be happening at very high temperature. So yeah, mention the use of the PCR in molecular diagnosis. So we can do the early detection of the AIDS, cancer, or gene mutation. Any genetic mutation can be found out, right? So this is the advantage of the PCR. So we can detect the AIDS, cancer or mutation causing in the mutation cause in the patient, right, which can cause cancer, or we can just detect the genetic mutation also, right? So these can be this technique will be helping in detection all these things. So this is the diagram. Role of transgenic animals in production of the biological products. So this is very easy. We know that production of the biological product is alpha 1 antitrypsin. No? So I'm sure you have seen. So we can actually form this drug alpha 1 antitrypsin, and it is used to treat the emphysema, which is the lung disorder, right? So alpha 1 antitrypsin, which is used to treat emphysema. Emphysema, you know, it's a lung disease. Next, it is also used to produce, you know, transgenic cow was produced, known as Rosie, and Rosie were producing the milk, human protein, alpha lactalbumin. Enriched milk. So that means this Rosie were producing the human protein alpha lactalbumin enriched milk. So this is also example of biological product you produce from the transgenic animals. Studying diseases, yeah, so we can use the transgenic animal to study the genes, which gene is responsible. We were actually inducing the mutation in particular gene, and we were seeing that why, if the mutation happening in this gene, what's the impact, what is the function of this gene, what is the impact of this gene. And in this way we can actually decipher a lot of the effect of the mutation, right, lot of the diseases. So we can study it very effectively, and it is used to detect transgenic animals, you know, their genes are used to study the diseases, study the diseases Alzheimer, or we are actually using them to study the diseases cancer, or we are using them for the different type of diseases, right, Alzheimer, cystic fibrosis, or many other kind of genetic disorders. We are using them, right, we are just changing their gene just to study the effect or how this gene is developing the particular structure, or if mutation happen in this gene, what will be the result, right? So that we can actually form the treatment, or we can study the disease, we can understand the disease, we can understand genetic disease or any disease, right, genetic disease or effect of the gene to develop treatment. Chemical safety testing. So when we talk about chemical safety testing, so that means what's happening here, we are suppose there are the drugs, right? So we just want to detect some side effect of certain toxin, right, which is present in the chemical. So we are modifying their gene, transgenic animals gene, in such a way that even a trace amount of chemical or toxin will actually make them so sensitive, will show allergies in their body, will show some kind of reaction. So they become so sensitive, these transgenic animal, we are just creating their gene in such a way, modifying their gene in such a way, they become so sensitive, even the trace amount of the toxins, than the normal animals, so that we can study the effect of the toxin very, very nicely, right? So we are doing that to study the effect of toxin by modifying their gene and making them so sensitive to these toxin. And this is the question where they have shown this diagram from NCERT, that is spermatogenesis. So they are asking you that what is this diagram representing? Name and describe the process depicted in the diagram which result in the development of spermatozoa. So you have to write down whole spermatogenesis, that, you know, spermatogonia, you will start with the spermatogonia, will undergo division and it will increase the size to form primary spermatocyte. Primary spermatocyte later undergo meiosis 1 to form secondary spermatocyte. Secondary spermatocyte undergo meiosis 2 to form the 4 haploid spermatids. And then spermatids undergo spermiogenesis or differentiation to form the 4 haploid spermatozoa. Right? So you have to write down the steps of the spermatogenesis here. Identify the cell where you are seeing a cluster of spermatozoa attached in the diagram. So they're talking about the Sertoli cell, where the spermatozoa are attached. Immature sperms taking nourishment. So what is the purpose of Sertoli cell? What is the function of the Sertoli cell? They provide nourishment to the developing sperm, right? That's all. So this is how that is what they are asking you in this question, right? So you have to write down that spermatogenesis will begin at the puberty in the human, right? In the male, and then it is because of the testosterone, and then you will write down that what are the steps of spermatogenesis, and that's all, right? So they are asking you this much only. And the last question, 33 question, is describe the life cycle of HIV from the time of its entry into the human body with till full blown AIDS sets in. So you will be writing this, that first of all you will be introducing what is AIDS, acquired immunodeficiency syndrome, who is the causative agent, HIV, human immunodeficiency virus. It is enveloped, so their body is covered, unenveloped virus, their body is covered by the membrane, right, by this proteinaceous membrane, capsid membrane, and they have lipid covering also. And then you will be writing about they will first of all enter the macrophages, which is the factory. So first of all they will be entering in the macrophages because they have the receptor, right? They have these macrophages has the receptor which is recognized by the receptor present on HIV virus, right? So where they will increase their number. So this is the factory, HIV factory, because it will increase their number. And simultaneously, when they are increasing their number, they will infect the T helper cell also, right? So simultaneously, they will be destroying T helper cell. So you have to write down all this in a point form, and then you have to just make this diagram very nicely to explain the effect of HIV, or how they are killing the T helper cell and how they are producing the new viruses using our DNA. So how they are introducing their RNA, viral RNA in our body, how using reverse transcriptase enzyme they are forming the viral DNA. Then using certain mechanism, they are just incorporating their DNA with our DNA. Using our transcription and translation machinery, they are forming new RNA, they are forming protein around it, and then they will be going out and taking lipid from our membrane. So full RNA virus, full AIDS virus is regenerated, and they are ready to affect the other T helper cell. So they will just decrease down the number of T helper cell, decrease our immunity, and then this person becomes susceptible for various other diseases, opportunistic diseases. Opportunistic means those pathogen which are generally not able to cause any disease in the healthy person, but when their immunity is decreased down due to HIV virus, now these bacteria easily can cause disease. They have got the opportunity. Oh, now the immunity is decreased down. Chalo, let's enter the body. Let's enter this patient body, right? And let's do some diseases. So you have to mention all these things here. Now, other in in the option, internal options, you have write the symptom of malaria. Reoccurrence of chills and fever after every 3 to 4 days, right? So we know reoccurrence of chills and fever after every 3 to 4 days. So you have to write down this in your answer and explain what caused these symptoms. So we know that the toxin hemozoin produced by these plasmid in the RBCs. So they eat hemoglobin and convert hemoglobin into the hemozoin toxin. And this toxin responsible, so when RBC burst, this hemozoin comes out and cause the fever and chills. Next, describe the different steps in the sexual mode of reproduction, which occurs in the female Anopheles mosquito, right? Sexual reproduction happen in the female Anopheles mosquito in the life cycle of the malarial parasite from the time of its initiation till where it's completed and ready to start a fresh cycle. So that means they're asking you to draw this part of life cycle. So gametocyte, female is large in size, female gametocyte large in size, male gametocyte comparatively smaller in size, right? So they will just, ingested by the mosquito from the patient body. Then in their gut, they will undergo fertilization. And, after fertilization form the zygote. After zygote, various steps happen. And then they produce a lot of these sporozoids. and these sporozoites get reserved in their salivary gland. So there's a mosquito, this Anopheles mosquito salivary gland is the reservoir of the sporozoites, which are the infective stage for human. So when they bite human, when this mosquito will bite other healthy human, this will be entered in the human and via blood reaches the liver cell. And what will happen in the liver cell? Obviously, they will proliferate themselves and then they will burst the liver cell, go to RBCs. In RBCs, they again undergo the division. They form a lot of the, pathogens, right? Lot of the parasites. And then they are eating our hemoglobin, forming the hemozoin, and then they are just bursting RBCs to releasing their daughter cells, and plus they are releasing the hemozoin, and that hemozoin will cause the chills and fever, right? So that means they are producing a lot of the copies by asexual reproduction in the human, and they are doing the sexual reproduction in mosquito. So they're asking you to draw this part of life cycle, right? So that's all. I hope that you have done well in your exam, and I hope you like the discussion. And we are done with the set 1 discussion, right? So in case you have any doubt, please write down in the comment box, and we are meeting again for the NEET preparation, right? So stay connected with our channel, stay tuned, and all the best, right? So just enjoy your time, or just have fun, just watch movies, whatever you want to do, or want to prepare for some exam, just do it, right? So if you need any kind of help, we are always there, right? So all the best, kids.